Hello dear viewers, welcome to Crown Biology classes. This is Rohit speaking and today I will be discussing chromosome packaging in the prokaryotes and chromosome packaging in the eukaryotes. In most viruses and prokaryotes, the single set of genes is stored in a single chromosome. That means haploid condition is present in most viruses and prokaryotes, and the single chromosome in turn contains a single molecule of nucleic acid. That chromosome is made up of single molecule of nucleic acid that could be either DNA or RNA depending upon the genetic material. Prokaryotes generally are devoid of histones. These histones are the characteristic features of eukaryotes genetic material. So prokaryotes do not have these histones, and in place of histones they carry proteins that are known as polyamines. In bacteria like Escherichia coli, the circular chromosome occurs as a circular unfolded chromosome, and that circular unfolded chromosome will be having a diameter of 350 micrometer. Such a large diameter of unfolded chromosome is there. So, in order to fit this into 2 micrometer space of the bacteria, the chromosome needs to be properly folded. This circular unfolded chromosome needs to be properly folded so that it can fit into 2 micrometer space. And it is quite challenging to fit the large DNA molecule into 2 micrometer space of bacteria. In the folded chromosome, the large DNA molecule in the E. coli is folded into 50 to 100 loops around the RNA core. As you can see in the diagram, A is the diagram of circular chromosomal DNA, and in B diagram what happens is formation of loop domains occurs. Formation of loop domains occurs around the RNA core, and in C supercoiled and looped DNA. B further supercoiled to form the supercoiled and looped DNA. Both proteins and RNA are part of this folded chromosome, and finally the chromosome becomes the supercoiled folded chromosome where the loops of DNA form negative supercoils. The loops of DNA can form negative or the positive supercoil, but in this case the loops of DNA will form negative supercoil, and it depends upon the amount of strain in it, and accordingly it can be negatively supercoiled or positively supercoiled, and the size of the chromosome becomes 2 micrometer. Such a small size becomes of the chromosome, and that is good enough to fit a bacterium. Starting with the chromosome packaging in eukaryotes, eukaryotes have a higher level of complexity and most eukaryotes are diploid, whereas prokaryotes and most viruses were haploid. That is, they consist of only one set of genes. But eukaryotes are diploid, that is, they have two sets of genes, and the interface chromosomes are generally present in the form of nucleosomes. Nucleosomes is nothing but a bead on a string model. Bead on a string model consists of a single strand of DNA or a chromosome, and the proteins are associated with it. However, during mitotic and meiotic phases, the DNA mostly occurs in highly condensed structure. During these stages, the DNA condenses to form highly condensed structure that is known as chromosomes. Chromatin is primarily made up of DNA and proteins like histone and non-histone proteins. Okay, chromatin is associated with histone and non-histone proteins. Histones are the basic characteristic feature of eukaryotes. Histones, there are five major types of histones, namely H1, H2A, H2B, H3, and H4. Histones are positively charged proteins as these histones contain 20 to 30% arginine and lysine amino acids, and these histones are not present in the prokaryotes. Polycationic charge side chain of histones are important because they interact with polyanionic negatively charged phosphate groups on DNA. As you can see in the diagram, there exists two types of interaction. The first one is hydrogen bonding and the electrostatic attraction between the polycationic charged side chain of histone and the polyanionic negatively charged phosphate groups. And these histones are found almost in all the eukaryotic cells, but there are few exceptions as well, such as sperms, where the histones are replaced by protamines. There are three levels of DNA packaging in eukaryotes. When chromatin is isolated from interface cells, when we do arrest the cell at interface cell of mitosis, and when the chromatin of that cell is examined, it is found to consist of series of ellipsoidal beads, and hence this model is called bead on a string model. This diagram below clearly illustrates beat on a string model. As you can see, the DNA is wound around histone proteins, making a structure that is known as nucleosome, and linker DNA is also associated with it. Starting with the first level of DNA packaging in eukaryotes, first one is beat on the string model. The majority of DNA in eukaryotic cell is packaged into nucleosomes. And what does a nucleosome consist of? A nucleosome consists of a core of 8 proteins and approximately 147 base pair of DNA wrapped around it. We know the structure of a nucleosome, that a nucleosome consists of 8 proteins, and the DNA is also wrapped around those proteins, and the length of that DNA is 147 base pair. The DNA most tightly wound around the nucleosome is called the core DNA. Alright, and it is wound around 1.65 times around the histone octamer. The linker DNA between the nucleosome has typically the length of 20 to 114 base pair. So you should keep this in mind that 147 base pair of DNA is wrapped around the 8 proteins, but the linker DNA which is present in between the nucleosome, it has a length of 20 to 114 base pair. So in any cell, stretches of DNA will be present, and those stretches of DNA will not be associated into nucleosomes, so those will be involved in other functions such as expression, replication and recombination. Expression, replication and recombination processes, the DNA is held together by non-histone proteins. As we know, the eukaryotic DNA is associated with histone and non-histone proteins. The nucleosome core consists of two molecules, each of H2A, H2B, H3 and H4. See, H1 is not associated here. H2A, H2B, H3 and H4, these are associated with 147 base pair of DNA. Importantly, H1 histone is not the part of the core nucleosome, but it is associated with complete nucleosome, where it is known to participate in higher order chromatin folding. So this H1 histone has its own role. It is known to participate in higher order chromatin folding, but the nucleosome core consists of only H2A, H2B, H3 and H4. The core histones have an amino terminal extension that is called histone tail. Core histones are H2A, H2B, H3 and H4. These histone tails lack a predefined structure. They don't have any predefined structure and they don't play a role in DNA association, but these are involved in another function. These tails are the sites for extensive post-translational modifications, PTMs. Post-translation modifications like acetylation, methylation and phosphorylation add serine, lysine and arginine residues respectively. The second level of DNA folding in eukaryotes is the 30 nanometer fiber. When H1 histone is added, then it stabilizes higher order structure like the 30 nanometer fiber. So the core histone consists of H2A, H2B, H3 and H4 proteins. And when H1 histone is added, it will stabilize the higher order structure like the 30 nanometer fiber. And the 30 nanometer fiber is very less compacted and it is less accessible to the DNA polymerases. And as a result of that, it is transcriptionally silent. Further, under 30 nanometer fiber, there are two models. First one is solenoid model and the second one is zigzag model. Solenoid model, from the name itself it is clear, it will be having a solenoid shape. In this model, the nucleosomes form a superhelix having 6 nucleosomes per turn. And the helix has a diameter of 30 nanometer with a helical pitch of 11 nanometer. Alternatively, the 30 nanometer fiber can have zigzag model. The array has a zigzag pattern of nucleosomes. From the name itself it is clear it will be having a zigzag pattern of nucleosomes, and the solenoid model is having the solenoid pattern of nucleosomes. The zigzag model requires the linker DNA to pass the central axis of the fiber, unlike the solenoid model, which do not require the linker DNA to pass the central axis of the fiber. So both the forms of 30 nanometer fiber, these can exist depending upon the length of the DNA. Linker DNA, as longer linker DNA prefers the zigzag model, as the linker DNA has to pass through the central axis of the fiber, so the longer DNA prefers the zigzag model. This is the diagram illustrating the solenoid model. As you can see, the helix has a diameter of 30 nanometer with helical pitch of 11 nanometer. And the nucleosomes form a superhelix having 6 nucleosomes per turn. So this is first model of the 30 nanometer fiber that is known as the solenoid model. We can easily compare solenoid and zigzag model in this diagram. The solenoid model is having the linker DNA which is not passing through the central axis of the fiber, but zigzag model is having the linker DNA which is passing through the central axis of the fiber, and both are the examples of the 30 nanometer fiber. The 300 nanometer loops, more folding of the 30 nanometer fiber is required for compacting DNA further, and it will result into large loops of 300 nanometer fiber, and they will be held together at a base by proteinaceous structure protein that is called scaffolds, and this is beautifully represented in this diagram below. Some of the proteins which are a part of the scaffold are topoisomerase 2, SMC, and cohesions are some of the proteins which are forming scaffold. Further, these 300 nanometer loops are condensed into 700 nanometer loops, and they are woven around to form the chromosome, the final structure which is 1400 nanometer in breadth. So this is the summary of what we have discussed just now. It is a short region of DNA double helix having a diameter of 2 nanometer. Then comes the beads on a string form of chromatin having a diameter of 11 nanometer. Then comes the 30 nanometer chromatin fiber of packed nucleosomes having 300 nanometer of diameter. Section of chromosome in an extended form is having the diameter of 300 nanometer, condensed section of chromosome having a diameter of 700 nanometer, and the entire mitotic chromosome is having the diameter of 1400 nanometer, as you can see in this diagram. Ending this video on a positive note, a little progress each day adds up to big results. Thank you for watching.